It is my distinct pleasure today to introduce our invited speaker, Dr. Michael Levin, and he is a world-renowned scientist with research spanning developmental biology, computer science, bioelectrics, and philosophy of mind. And Dr. Levin is the Vannevar Bush Distinguished Professor of Biology at Tufts University. He is an associate faculty at Harvard's Wyss Institute and the director of the Allen Discovery Center at Tufts. He has published over 400 peer-reviewed publications. His graduate research was published in Cell Journal in 1995, and then it was chosen by journal Nature as a milestone in developmental biology in the last century. And I can go on and on, but instead, I just want to say that Michael is one of the most brilliant scientists and one of the best speakers I have ever had the privilege to meet. So please join me in welcoming Dr. Levin.
Thank you so much for that extremely kind introduction. I wish I was there in person to meet with you again. But anyway, thank you for having me here to share some ideas. What I'm going to talk about is endogenous bioelectrical networks. And I'm going to argue that this is not simply another layer of physics that we need to understand, but actually a very profound interface to the intelligence of the body, which has many implications for regenerative medicine. And if you're interested in downloading any of the primary papers, the data sets, the software, everything is at our lab website here. And this is my personal blog around what I think some of these things mean. I'd like to begin by considering the end game of our field. That is, where is all this going? Imagine with something I call the anatomical compiler. Someday you will sit in front of a computer and you will be able to draw the plant, animal, organ, biobot, any living structure, whether already existing or imaginary, any living structure. And what this thing will do is compile your description into a set of stimuli that have to be given to cells to get them to build exactly that structure. Now, why we need it is quite obvious, because if we had the ability to communicate our goals to groups of cells, we would automatically end birth defects, we would be able to repair traumatic injury, reprogram cancer, reverse aging and degenerative disease. All of these things would be solved if we had the ability to tell groups of cells what to build, healthy new organs, living machines, whatever. And what's important to note is that this is not some kind of a 3D printer. The goal is not to micromanage the position and the gene expression of cells. This is meant to be a communications device. It's a translator between our goals as engineers and workers in regenerative medicine to those of the cellular collective. But how to do this? Why don't we have this? You know, genetics and biochemistry and molecular biology have been going strong for decades. Why don't we have anything remotely like this? Because the situation that we are in is this: we are very good at now as a field at manipulating cells and molecules and knowing which molecular machinery binds and acts on which other kinds of molecules. But we're still a very long way away from control of what we're really interested in, which is large-scale form and function, let's say restoring somebody's entire limb. And actually, we're quite far, in most cases, from biomedical treatments that actually fix things in a permanent way. We don't have very many of those. And that's because today, I would claim that biomedicine is largely stuck at the level of hardware. In other words, it is where computer science was in the 1940s and 50s. This is what programming looked like in the early 1950s. You can see that in order to get this computer to do something different, she's rewiring the hardware. She's physically interacting with the hardware, and this is where modern molecular medicine is today. All the exciting things are at the level of genomic editing, protein engineering, pathway rewiring. It is all at the level of the hardware. And I think in order for biology and medicine to reap the kinds of benefits that we have seen in the information technology revolution, we would have to move from an exclusive focus on our biological hardware to understand the software of life. And what I'm going to argue today is that largely speaking, the most exciting part of that software of the living material is its intelligence, its multiscale problem-solving capacity. So today, I would like to give you these 4 points. First, I'm going to show you that bodies are constructed of a multiscale competency architecture, basically agential systems at multiple scales that solve problems in different spaces. I'm going to argue that definitive regenerative medicine is going to require exploiting the collective intelligence of groups of cells, communicating anatomical goals, collaborating with their competencies, not micromanaging them. And most appropriate for this venue, I'm going to show that endogenous bioelectrical networks in all tissues, not just brains, are a very highly tractable interface for this kind of control of growth and form. Tools are now coming online to read and write goal states into the living medium of the body, which is, I think, going to be the future of regenerative medicine. And this, of course, will give rise to some examples that I will show you of applications in birth defects, regenerative repair, and cancer. And the whole message of the talk, if I had to say it in one sentence, is basically this: that the bioelectricity serves as the cognitive glue for the collective intelligence of bodies, with all the implications that has. So first, let's just consider what we are all made of. This is the kind of material that we are all constructed of. Now, this is a free living organism called the lacrimaria, but you get the idea. The cells are incredibly competent beings on their own. There's no brain here. There's no nervous system. There's no stem cells or cell-to-cell communication. But this little creature in one cell is doing everything it needs to handle its physiological, anatomical, and metabolic requirements. If you're going to engineer with a material like this, you have to work very differently than engineering with passive matter like wood and metal and things like that. All of that is described in this review of synthetic morphology with these agential materials. So we know that cells are very active and competent, but in fact, even below the cell level, the molecular networks inside of cells are themselves capable of, for example, at least 6 different kinds of learning. They can do habituation, sensitization, and even Pavlovian associative conditioning. And in our group, so all of that is described here, but in our group, we're building devices to train the molecular pathways inside of cells with all kinds of cool applications to, for example, drug conditioning and things like that. So really what you see is that it's not only the brain and nervous system that are capable of learning and decision making. This kind of thing goes all the way down, and we could spend hours on many examples. I don't have time to do that here, so I will just move on with the general point that we as humans, because of our own evolutionary history, are reasonably good at recognizing intelligence of medium-sized objects moving at medium speeds through three-dimensional space. Birds, primates, things that, maybe a whale or an octopus. We can sort of recognize their ability to navigate three-dimensional space. We can see that they solve problems and that they're intelligent. But biology has been doing this long before brains and muscles appeared. Living systems live in all kinds of exotic spaces that are very difficult for us to visualize, but they make decisions and they traverse these spaces in goal-directed ways. They live in transcriptional space, a very high-dimensional space of gene expression states. They live in physiological state space and anatomical morphospace, the one that we'll talk about the most today. So this idea of embodiment, of being able to perform this loop of sensing, decision-making, and then action takes place in many different spaces. And today we will talk about how living systems navigate the space of anatomical possibilities. So the first thing to ask is, where do anatomies come from in the first place? So here's a cross-section of a human torso. You can see enormously rich structures. Everything is typically in the right place, the right size relative to the right neighbors. Where does this pattern come from? Where is it written down? Because we all start life like this, as a bunch of embryonic plastomeres. Where does this pattern come from? And people will initially, of course, say, well, of course, the DNA, it comes from the genome. The human genome tells you what to do. But we can read genomes now, and we know that what the DNA specifies are proteins. What we're interested in is a three-dimensional anatomy. How do cell groups know what to make? How do they know when to stop? Because this structure is no more directly in the DNA than the structure of a termite colony is in the genome of the termites, or the particular pattern of a spider web is in the DNA of the spider. All of these are the outcome of physiological software running on the hardware that's encoded by the genome. So we have to understand how do cells know what to do? How does this hardware enable that? And how do we induce them to repair when things are missing or damaged? And actually, a large chunk of our lab is working on this. Can we actually get them to build something completely different? And I'll show you just a brief glimpse of it in this talk. So I've said a number of times that the living material has intelligence baked in at multiple levels of organization. And what I mean by intelligence is this definition by William William James. I don't mean that it's a human level metacognitive ability to know what you're doing or anything like that, but simply the functional ability to reach the same goal by different means. In other words, to adaptively navigate a landscape in a goal-directed way with some degree of ingenuity to get your goal met when things change. So let's take a look. What kind of collective intelligence do swarms of cells deploy? Well, the first thing we know is that morphogenesis is incredibly reliable. So almost all of the time, this process gives rise to the correct target morphology. But that is not why I'm calling it intelligence. It is not reliability. It is not the emergent complexity. That's not it. Those are cheap and easy. You don't need intelligence to have those things. What's actually the reason I call it intelligence is specifically because of its problem-solving capacity in navigating that space when things change. What kind of things? Well, the first thing you can do is for most kinds of embryos, you can cut them into pieces. You don't get half bodies. You get perfectly normal monozygotic twins, triplets, and so on. And that's because from different starting positions, whether this normal position or this injured position, these systems can navigate anatomical space and still reach the correct ensemble of goal states that they're trying to get to, despite pretty radical deformations. And they can avoid local minima of all kinds, and they still get to where they're going from different starting positions. So that's the beginning. By the way. This is not only for embryogenesis. In fact, I think embryonic morphogenesis is just a special case of this anatomical homeostasis property. So some animals do this as adults. So here is an axolotl. This amazing amphibian can regenerate its eyes, its jaws, its limbs, its spinal cord, portions of the brain and heart. But what happens in the limb is that if you amputate anywhere along this path, the cells will immediately spring into action. They will build very rapidly, and then the most amazing part of this is that they stop. When do they stop? They stop when a correct salamander limb is complete. So this is actually a homeostatic loop. When they sense that they've been deviated, so the error is high, they will work to reduce that delta. Okay, they get back. They're traveling back to that region of anatomical space, and as soon as they reach it within some very small tolerance of error, then they stop. So this is a homeostatic process. By the way, regeneration is not just for amphibians and the worms I'm about to show you. Some human organs do it too. The human liver is highly regenerative. Even the ancient Greeks knew that. I have no idea how they knew that, but they did. Deer are an adult mammal that every year regenerates huge amounts of bone, vasculature, innervation, up to 1.5 centimeters of new bone per day during this process. Okay, and human children below a certain age regenerate their fingertips with a very nice cosmetically perfect outcome. So this is quite a distributed kind of capacity to restore from damage. But it's not all about restoring from damage. It's much deeper than that, because here's another interesting experiment done in the 50s. If you take a tail of an amphibian like this and you surgically attach it to the flank between the two limbs, what you will see is that over time it becomes it metamorphoses. It becomes converted into a limb and a structure that belongs, at this new location. Now, the amazing thing about this is that even the tip of the tail, which are tail tip cells sitting at the end of a tail, so locally there's nothing wrong, they're perfectly correct, but they become fingers. The whole thing, even the regions that locally have no issue, become remodeled. Because what you have here is a kind of top-down control. The global body plan dictates local cell and molecular states, converting them, and by using the plasticity that's inherent in this creature, converts them to a new structure that's more suitable for the large scale. And actually, there's even some human data on this now in terms of transplants, people getting arm transplants or seeing remodeling of their arms to better suit the rest of the body and so on. So this is a remarkable ability of multiscale intelligent systems, just like us. You know, when you have thoughts and goals in your mind that are very abstract, you might have career goals or social goals or whatever. In order for you to move around and to walk to execute those goals, in other words, voluntary motion, those very abstract high-level goals have to be converted in your body, converted to ions moving across your muscle membranes, literally mind over matter. And that is exactly what's happening here. Large-scale collective goals of a system trying to be a proper amphibian is filtering down to the molecular cues that have to tell individual cells what they're going to be. So this is a fundamental feature of a multi-scale cognitive system. And then you have things like this, which again are showing us the flexible navigation of anatomical space. Here's a normal tadpole. So here's a brain, the gut, the nostrils, the mouth, the eyes. And these things have to drastically rearrange their face to become frogs. Everything has to move around. And it used to be thought that these kinds of movements were hardwired. Somehow the genetics just makes everything move it to the right amount in the right direction. Well, we decided to test that, and that's very important. Any claim of intelligence in any system has to be tested experimentally. You have to show the problem solving. So what we did was create these so-called Picasso-like tadpoles. Basically, everything is scrambled. The eyes on top of the head, the mouth is off to the side, everything is in the wrong position. And we found that these give rise to actually quite normal frogs, because all of the organs don't just go in the same paths, they go in novel paths as needed to give you a correct frog, and then they stop. In fact, sometimes they go too far and they actually have to come back. So what the genetics actually is giving you is hardware that can execute a highly flexible, context-sensitive goal-directed program of reaching a specific location in anatomical space, and it will do it in different ways depending on what's going on. And so we need to now ask the question, how does it know what the goal is? How do any of these systems store the set point? Right? Any homeostatic process has a set point, and they have to be able to store that somehow, and then compute the delta and try to reduce that error. So how do they do this? I'm about to show you. But before that, one last thing. What I'm showing you here are examples of systems that are navigating space to reach their default goals. But what happens if that goal is completely unreachable? What if it's impossible to reach? Well, one thing biological systems are very good at is deriving novel goals. If you can't reach your original goal, come up with a new goal. And so what I'm showing you here is just one example of many that I could tell you about. This little creature, it looks like a primitive organism I got from the bottom of a pond somewhere. You can see it navigating around, and close up, this is what it looks like. It has these little hairs on the outside surface. They're cilia, and that's how this little guy moves around. You might guess that it's a primitive organism with a primitive genome. If you were to sequence the genome, what you would find is 100% Homo sapiens. Unedited human genome. This comes from adult cells, from adult patients, taken from tracheal epithelia. When extracted from the patient, these cells self-assemble into this little self-motile little living creature, which has a lifestyle completely different from that of the human that it came from. It looks like no stage of normal human development. It has some interesting features. It expresses about 9,000 genes, in other words, half the genome, differently than the way it did when it was in the body. Radically remodels its transcriptome. It has 4 specific behaviors that it can do, and you can build a little ethogram of it. One cool trick that they can do, if you plate a cluster of human neurons here and put a big scratch through it, they will assemble into something we call a superbot. So there's, I don't know, about a dozen of these things lit together. And what you can see is that over about 4 days, they knit together across the gap. They heal the neural wound. So who would have thought that your tracheal epithelial cells have the ability to make a self-motile little creature that also can heal your neural wounds? So lots of biomedical implications here, but fundamentally what you're seeing is that an unedited genome, no Yamanaka factors, no synthetic biology circuits, no genomic editing, no weird scaffolds or drugs or nanomaterials, just the cells themselves. If they can't do the normal thing that they do in the body, they will find something else to do, and often it is quite coherent and quite remarkably complex. And so this is yet again another skill. Besides reaching your familiar goals, is finding new attractors in the space of different ways to be alive. And so fundamentally in our group, what we've been studying, and this is described, some of it is described in this paper. What we've been studying is the ability of networks of living cells to use various principles that are, interestingly enough, now studied in machine learning and computational psychiatry and things like that, to create networks that actually interpret the information they're receiving from the genome, not follow it as a mechanical device, but actually creatively interpret them as cues. And this bow tie that occurs when individuals compress down everything that the lineage has learned over the evolutionary time into a single generative seed, like an egg, and then it has to be creatively reinterpreted during morphogenesis, maybe as the normal embryo, but maybe like an anthrobot or like many other things. So we really have to ask, in all of these examples that I showed you, what really underlies the ability of groups of cells to remember their goals? What allows them to reduce the delta between what's going on now and what they think is the target. What allows them to interpret this information, this environmental and genetic information, creatively to solve novel problems? What's the machinery that does it? And it turns out that we can get a lot of cues from that from looking at how the brain does things. And what's happening in the brain is that you have some remarkable machinery that allows the individual selflets. So here's a single, for example, a single human extended over time, made up of a bunch of slices, time slices of being. And at every slice, what the nervous system has to do is create, basically confabulate a story, a self model of and a model of the outside world from memory engrams, from messages left by prior versions of yourself. In other words, memory engrams left in your brain and body have to be continuously interpreted in a way that executes on various adaptive goals, right? And so the way the brain does this is through electrical networks. And so of course you have these cells that have neurons that have ion channels in their membrane. They have electrical synapses to other cells, and it forms this amazing electrophysiological network where now, for example, here this group tracked the electrophysiology of a living zebrafish brain as the zebrafish thinks about whatever it is that fish think about. And the idea is that if we could read out all this electrophysiology and decode it, then you would have access to all the memories, the goals, the preferences, the capabilities of the agent, right? The cognitive content is encoded in this electrophysiology. Remarkable architecture. But the thing is, evolution discovered this a very long time ago, long before brains and nervous systems appeared. So back around the time of bacterial biofilms, evolution was already using electrical networks as a kind of cognitive glue to bind individual competent subunits, microbial cells, into a large-scale collective that can do things and that knows things that individual cells don't know. And there's beautiful work by Agorol Suel out of UCSD on that whole evolutionary story. But for us, what's important is that every cell in your body has ion channels. Most of them have electrical synapses, or these gap junctions to their neighbors. And so for about 20 years now, we've been working to basically take everything that's been done in neuroscience to understand the cognitive glue, the electrophysiology that makes you and I more than the sum of our neurons, and do the same thing for the rest of the body. That is, read and decode the electrical activity of the body cells and ask, how do these electrical networks help cells navigate anatomical space, the way that brains help you navigate three-dimensional space. And the deep lesson that we've learned is that almost everything from the behavioral and cognitive neurosciences can be translated into developmental biology. If instead of, you can take almost any paper in neuroscience, and anytime it says neuron, just say cell. Anytime it says millisecond, you say minutes or hours, and a couple of other small changes, and you've got yourself a great developmental biology paper. So there's huge symmetry between the formation of the body and the formation of the mind, because the mechanisms are evolutionarily homologous, not just analogous, but actually homologous. And so we've developed tools to read and write the electrical patterns that are driving this collective intelligence. So using voltage-sensitive fluorescent dyes and new technologies like fluorescent lifetime imaging, we can see all the electrical conversations that the cells are having with each other. We make computational models of what's going on. And more importantly, we can, oh, by the way, here are some examples of video of this kind of thing in the real system. So here's a frog embryo putting its face together, and you can see there's a lot going on, but here's one frame from that video. What you can see is this is a pre-pattern. We call it the electric face. It looks like a face. You can already tell. Here's where the eye is going to be. Here's where the mouth is going to be. Here are the placoid structures. This is the electrical pattern is a subtle scaffold that controls the gene expression that is going to regionalize the face and result in the normal craniofacial anatomy. Not only is the bioelectrical pattern binding individual cells towards large-scale organ-level outcomes, but actually does the same thing on a multi-embryo scale. So for example, if I poke this embryo here, you can see that there's a calcium wave that within 30 seconds all these neighbors find out about it, right? And you can see the same thing here. After I poke that one on the left, all the neighbors find out about it. So bioelectricity is a multi-scale kind of system that allows collective dynamics. And by the way, the improvements in this field have been remarkable. I kind of like this. The example from astronomy, in 1996, this is what we had for Pluto. This is what Pluto looked like in 96. By 2002, it looked like this, and by 2017, it looked like this. You can see actual features. And the same thing is true. This image of the electric face was made in 2012 by Danny Adams. Here's what the eye spot looked like that. Here's what it looks like now with modern film imaging, and this is the work of Patrick McMillan, who's really revolutionized a lot of our ability to read these electrical signals. But reading them is just the first start. You really have to be able to rewrite the patterns. That is, not just listen to what the cellular collective is doing, but actually communicate with it and write patterns back into it. We don't use any applied fields, so there are no waves, frequencies, electromagnetics, nothing like that. What we're doing is using the tools of neuroscience to hack the interface that cells normally use to control each other. That is, we can control the gap junctional connectivity between cells, so the topology of this tissue network, and we can turn channels on and off using traditional methods, using pharmacology, optogenetics. We can also make modifications in the channel structure and so on. So basically, we control the synaptic plasticity and the intrinsic plasticity, just like neuroscientists do. And now we can write new messages into the system, and I'm about to show you what happens. But the first thing I want to show you is some amazing work by Patrick, just illustrating what it means when cells are controlling each other using this electrical interface. So I'm going to play this video in a second, but notice what happens. Here's a single cell. Here's a bunch of cells here. The color represents voltage. Okay, so this is voltage imaging dye. The color is voltage. This cell has a very different voltage than the rest of them, and what's going to happen in this next frame is it just touches right here, just barely touches. So you can see what happens. So here it is, it's crawling along, minding its own business. This guy reaches out and touches it, boom. And now it's got the same voltage, and once it does, it joins this collective. So you can see here the tiniest of contacts is able to basically take over and impose a new bioelectrical pattern with consequences for cell behavior. So this is the native version. This is what natively happens. So let me show you what happens when we try to do the same trick. So I showed you that the position of the native eyes in the head is set by this particular voltage pattern, the eye spot. And what we can do is impose that pattern elsewhere in the body. And we do that by injecting ion channel RNA, encoding potassium channels that will set up that same voltage spot. If you do that, let's say in the gut, the cells are perfectly happy to do what the message says and build an eye. If you take cross sections through that eye, you find lens, optic nerve, all the things that belong inside an eye. It's a very well-formed eye. Now notice a few interesting properties here. First of all, I'm showing you that the bioelectric pattern is instructive. In other words, by giving a new message to the cells, I can get them to grow a brand new structure. This is not just making a defect. This is not just preventing some kind of passive or permissive kind of pathway. This is instructive for the organ. The second thing is it's incredibly modular. The eye is a very complex organ. We don't know how to build an eye. We don't know how to control all the gene expression that needs to change, all the position of the stem cells and all the different structures. We have no idea how to micromanage that. All we provide is a very high-level subroutine call that says build an eye here, and the system does all the rest. Again, this is the hallmark of a cognitive system, because just as when I talk to you, I don't need to worry about all the molecular synapses in your brain doing the right thing so that you can understand me. You will do that yourself. All I need to do is to provide the stimulus via a very narrow linguistic interface. You will handle all the biochemistry of the brain to form memories and to have opinions about it and so on. And so, and then two more quick things. One is that, if you look at the developmental biology textbook, what it had said was that actually only the cells up here in the anterior neuroectoderm are competent to become eye. And that's because people have been prompting it with a PAX6 gene, which is called the master eye gene. And it's true, in frog, the master eye gene only induces eyes up here. But that's because they were using the wrong prompt. It's not the cells that were not competent to make eye. Actually, almost any cell in the body can make this eye. It's because we as engineers were using the wrong prompt. The competency limit was on our end, not on the system's end. So it becomes very important to understand how to communicate convincing messages to the system in order to overcome certain limitations. And then the final thing that's pretty cool is, which will be important for the next slide, is this. If we take a section through one of these eyes, what you often find is that there's only very few cells that we actually targeted. The blue ones are the ones we injected. But all this other stuff was never injected by us. Instead, what happened is that the blue cells recruited their neighbors to become part of this eye. Again, competency of the material itself. We didn't teach them to do that. We have no idea how to make them do that. They already do that. As once they become convinced that they need to build an eye, somehow they can tell there's not enough of them, and they recruit a bunch of their wild-type neighbors to help them do that. And so this, again, brings up this point, which is important for therapeutics, which is that if you're going to try to change the goals of this cellular collective, you need to be convincing. You need to get buy-in of the system. This is not micromanagement. This is not rewiring. You have to get the system to take on the new goal. And often we fail at this. And what I'll show you is this. Oftentimes when we inject embryos, look, here's 1, 2, 3, 4, 5. Well, this is the native eye. So you've got these 4 ectopic eye spots. This is gene expression for RIX1, which is a very early eye marker. And if you were to look at this, you would say, oh, this embryo is going to have 4 extra eyes. Fantastic. But in fact, oftentimes you get none or sometimes 1, because what's happening here is that the rest of the cells resist. There's a cancer suppression mechanism that says, okay, if I'm your neighbor and you have some kind of weird voltage that is saying to be eye, you're telling me to be part of your eye, so you're trying to recruit me to be part of your eye, but I'm also going to try to recruit you to whatever we were doing before, to be skin or muscle or gut or whatever. I'm not just going to let you take me over. And so sometimes the one pattern wins, sometimes the native pattern wins, and it's really a battleground of models so that the cells have about what should be located in this region. We know they know what it should be, like that example I showed you with the tail being grafted to the middle of the flank. The system has a very clear model of what the correct pattern is, and if you're going to override that, you need to be convincing about it. And so this is very important. Learning to speak the language that really activates these kinds of things is therapeutically what we're looking for. So a couple of other examples in regeneration. So frogs, unlike salamanders, do not normally regenerate their legs as adults. So here, 45 days later, you normally get nothing. We created a cocktail that drives a message. This is an ion modifying cocktail that drives a particular message that says, go to the leg building path, not the scarring path. And sure enough, it activates downstream pro-regenerative genes like MSX1, and then eventually in 45 days, you've already got some toes, you've got a toenail, and eventually a pretty respectable leg. It's got the right shape, it's touch sensitive, it's motile, it's functional. One of the most amazing things about this is that the intervention, which we deliver by a wearable bioreactor, is only there for 24 hours. After you've given your message over 24 hours, you then get 18 months, a year and a half of leg growth, during which time we don't touch it at all. So this is not about micromanagement. This is not about telling the stem cells what to do. This is about convincing the tissue at the very beginning to head down one particular region of anatomical space rather than another, and then we take our hands off the wheel. That is what we are looking for, is triggers of complex downstream behavior, stimuli, not micromanagement. And I have to do a disclosure at this point. A company called Morphosceuticals is a spin-off by David Kaplan and myself, where we're going to try to move this technology to mammals, of course, hopefully someday humans, in building these wearable bioreactors to deliver the most convincing message that we can in the form of electroceutical cocktails sitting in the gel that is inside of this bioreactor that is going to support the growth. The next example I want to show you has to do with the ability to rewrite pattern memories. So what I've shown you so far is the ability to hijack the electrical system to insert messages to change the bioelectrical system. And I want to drill down on this example for a couple of minutes to show you that what we are actually doing is rewriting native memories. So this animal is an amazing creature called a planarian. These flatworms, they have one head, one tail. And we've been studying their properties of regeneration. So in planaria, if you cut them into pieces, every piece gives rise to a worm. And in fact, under normal circumstances, extremely reliably, you get worms with one head, one tail. And so we asked, why is that? How does this middle piece, if you chop off the head and the tail, how does this middle piece know which wound gets a head and which wound gets a tail? Maybe why don't they both get heads? And it turns out that there's a voltage gradient, which we can see here, that as we found out, says one head, one tail. And so when you do that, you very reliably get this. So what we were able to do is to using drugs that modify native ion channels and pumps. All it takes is about 3 hours. So the whole process of regeneration takes about 8 to 10 days. But in the first 3 hours, what you can do is you can change this pattern to be like this. So 2 heads here. And if you change this pattern, at first nothing happens. In an intact animal, they look perfectly normal. Anatomically, 1 head, 1 tail. Molecular biologically, the head marker is only in the head, not in the tail. You have no idea that anything is wrong with this animal until you cut him. If you cut him, taking this middle fragment, he will do what the pattern says, and he will build to a 2-headed form. So what's happening here, so again, this map, this bioelectrical map, is not a map of this 2-headed animal. It is a map of this anatomically normal 1-headed animal. The internal model of what a correct planarian should look like, the goal state, the set point, the representation of correct anatomy is encoded by this bioelectrical pattern, and it can be different than the anatomy. The anatomy is 1-headed, but already the pattern of what I will do if I get injured in the future, in other words, a counterfactual memory. So again, the beginnings of this amazing thing that evolved into our brain's ability to do mental time travel, right? To imagine scenarios that are not true right now. This is, I think, the basal version of this, the very simple version of this in the somatic intelligence, the ability to hold at least two different models of what the future should look like in the same body, in exactly the same body. Now, I keep calling it a memory in part because it has all the properties of memory. It is rewritable, as I just showed you, but it's also long-term stable. So once we make these 2-headed worms, we can continue to cut them again and again and again with no further treatment. You don't need to do anything else. These fragments will continue to generate 2-headed animals as many times as you cut them because the memory circuit holds. Once you've changed the set point to say 2-headed, that is what it will hold, and it will generate new individuals like this that have the same commitment to a 2-headed body plan. And here you can see this is the video of these little guys moving around. One more piece of data, and this is something that's actually not published yet, but I thought I would show it to you. The work of Johanna Bischoff, again to talking about this business of being convincing when you're trying to change the goal state of a collective system. What she did here was we wanted to see if we could transplant the 2-headed memory. So we took a piece of tissue from a 2-headed worm, surgically transplanted it into a normal 1-headed host. By the way, if you want to irradiate it in the meantime to kill all the stem cells, it's fine. It doesn't change the outcome at all. What happens is that a percentage of these guys, and here's roughly what it looks like. A percentage of these different fragments will give rise to 2-headed animals. So even up here, even here and here, which do not contain the implant at all, will give rise to 2-headed animals. Now, what's really interesting about this is that it's a tiny piece. You know, why would okay? So this piece believes it should be 2-headed, but all these other cells have a perfectly normal pattern. Why are they listening to this one piece? And it's we still really don't understand very well what it is that makes certain messages convincing to their neighbors. How come in 20% of the animals, or close to 10% of the animals here, the message that's coming from here actually takes over these other pieces? And it's not just about the number of heads, it's also, for example, about head shape. So you can take this species that has a nice triangular head shape and cut off the head, perturb the electrical network topology, and they will happily make flat heads like a P. felina. They will make round heads like an S. mediterranea. And of course, they can make the normal ones. Not just the head shape, but the shape of the brain and the distribution of stem cells is just like these other species. Between 100 and 150 million years of evolutionary distance between this one and these, and the same hardware, the standard genetically unmodified hardware, is just as happy to make 2 heads or heads belonging to other species. It can live in other attractors of this anatomical space. It can visit these other attractors where the other species live there stably all the time. You can even go further than the standard other species attractors, and we can make crazy looking things like cylindrical planaria and sort of hybrid forms and these weird spiky things. So the morphospace is very large and very weird, and you can make all kinds of the same hardware it's happy to build. Notice that we are not the only bioengineer. There is, for example, this wasp, okay, which is a non-human bioengineer. And this process of acorns making oak leaves, incredibly reliable, and people will say, well, this is what the oak genome knows how to do, is knows how to make these leaves. But that's not all. If you know the right prompt, and that's what the wasp is doing, is putting down some kind of prompt onto the tissue, they get the plant cells to build something completely different. I mean, who would have even guessed that the cells that build this nice flat green thing are capable of building something amazing like this, spiky and round and red and yellow? The material is highly competent, and we shouldn't let the reliability of the native outcome lull us into a false sense of stability against what can happen if you know what the right prompt is. It took this wasp millions of years, presumably, to figure out the right prompt by evolution. I hope we can do better, and to that end, we're building quantitative predictive models so that we can actually infer interventions and study these dynamics in a way that allows us to provide very highly convincing targeted signals that have very complex outcomes all across the layers of organization, from the molecular networks that specify ion channel expression all the way up to tissue dynamics, and then eventually algorithmic representations of head and tail decisions and things like that. And in the last few minutes, I just want to show you a quick example of a successful version of that, which is fixing birth defects. So here's a normal tadpole brain. So here's the forebrain, midbrain, and hindbrain. You can see it has very specific structure. All sorts of teratogens, for example, nicotine, alcohol, all kinds of things, will disrupt this normal structure. So here's a severe birth defect. And what we try to do is to create a computational model, and this is the work of Alexis Paitaka and the Vaibhav Pai in my group, to make a computational model of this process and be able to ask, what channels do we need to open and close to get back to the correct state? And I'm going to show you here just one example, which I think is the most the most difficult and I think most profound example. So here is a normal tadpole brain. This is a brain of a tadpole bearing a dominant notch mutation. Okay, so this is notch is a really critical neurogenesis gene, and when notch is mutated, you can see there is no forebrain. The hindbrain and midbrain are a bubble of water. They just lay there. They have no behavior, profoundly disabled animals. But we asked the model, okay, we can see how the bioelectrics that dictate brain shape and structure change after this mutation. How do we get back to the correct pattern? And the model predicted this one particular channel called HCN2, that if we were to open it, it has this really interesting property of sharpening electrical gradients. And it said that the model said that should get us back to normal. And so we did that either by introducing new HCN2 or using human-approved HCN2 openers. They happen to be anti-epileptic drugs in use in human patients. About half the animals had a perfectly normal brain. They had a normal brain structure, they had normal brain gene expression, and they had learning rates indistinguishable from controls. In other words, they got their IQs back, even though they're bearing this really nasty notch mutation. So at least in some cases, and I'm not claiming all cases, but at least in some cases, hardware defects can be ameliorated in software. In other words, transient manipulations of the electrophysiology can overcome what are otherwise really debilitating genetic diseases. And again, it remains to be seen how broadly this works. But we now have a roadmap for achieving this using a computational model to tell you exactly which drugs to use. So this is the kind of system that we're building, and you can play with an early version here, where you would start with data on the incorrect, let's say a tumor state or something, and the correct pattern you would like it to have, and the system would actually figure out which channels or pumps need to be targeted. That tells you exactly which drugs, and something like 20% of all drugs are ion channel drugs, so that there is an incredibly large toolkit of electroceuticals out there. We're just waiting to be repurposed for these goals, and it will suggest which ones to use. So this is a very bare bones system. Lots more needs to be added, but you can already see what's happening here. Okay, and then the final kind of chapter of the story, I just want to mention this idea that we talked about reaching goals. And we talked about the ability of cells as a collective intelligence to do that in various spaces. But I just want to mention the other interesting thing about this is the plasticity of the size of the goals. So individual cells have tiny little metabolic goals and physiological goals and things like that. But during evolution and during development, groups of cells can maintain enormous, grandiose goals, like this construction project in anatomical space here, where no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely knows, and you know that because of this homeostatic process here. And so that's a scale up of the system's intelligence in being able to pursue larger and more complex goals. But that process has a failure mode. That failure mode is called cancer. Basically, what happens is that when individual cells disconnect electrically from the collective, and that can happen by the function of oncogenes, but also by stress and by environmental toxins and so on. They basically revert back to their unicellular amoeba-like state. They go back to their tiny little goals. The border between self and world shrinks. Cancer cells are not more selfish than other cells. They just have smaller selves because now they just treat the rest of the body as external environment. The boundary between self and world has now shrunk. Instead of being this is one giant self pursuing giant goals, this is now back to their ancient lifestyle. And so that suggests an interesting therapeutic for cancer. Well, first, detection modality. So we can see the voltage change as these cells. This is a tadpole injected with a human oncogene. They will make tumors. They will metastasize. But long before any of that, you can already see the cells disconnecting. So you can see where the tumor is going to be. We are sort of working on, and this is an artist's rendition. This doesn't exist yet, but this is what we're working on: augmented reality goggles, so that when you're doing surgery with a voltage dye, you can immediately see, okay, here are the borders of the tumor, and here we better get this thing. So you can immediately see what's going on. But more than just being able to diagnose, what we found is that if we forcibly reconnect these cells back to their collective, we don't not kill them, not fix the genetics, but just we're using ion channels, reconnect them to their neighbors. You can actually get the individual cells to forget all about their tiny local selfish goals and go back to the large-scale morphogenetic set point. So here, this is the same animal. The oncoprotein is blazingly expressed here. In fact, it's all over the place. But look, there it is. But there's no tumor. Okay, and if you were to just sequence the cells, you would make the wrong prediction. You would say, oh yeah, there's the KRAS mutation. There's definitely going to be a tumor there. But that's not what drives. It's not the genetics that drives. It's the physiological decision making. And if these cells are connected to their neighbors, they will work on nice skin, nice muscle, all the other stuff, as opposed to going off and making a metastatic tumor. We are now moving all this stuff from frog to humans. So this is the first Juanita's first paper. In glioblastoma, and then we have some other things in colon cancer that are pre-printed and working their way through publication. But we think that along with regeneration and birth defects, cancer is a major target of this by developing tools to reconnect cells to their large-scale network that remembers adaptive goals as opposed to single-cell behavior. And the final thing, just to touch on, is aging. So if you're interested in aging, we have a couple of papers looking at how these bioelectrical patterns actually change over time. And there are a number of interesting things that happen to them over time that we think result in an aging phenotype. And we think this is quite actionable for longevity therapeutics, that we can sharpen these patterns and hopefully delay and maybe even reverse aging. So I'll just wrap up here and say that the most important thing we're seeing now is that biological bodies, are multiscale agential materials. There is problem solving, there are agendas, there is learning capacity, and there are defects in all of those properties that can come up at many levels of organization, and that we need to learn to communicate with all of these different levels, not just the molecular level of receptors and transcription factors and things like that, but the higher levels that lie between the level accessible to the molecular medicine groups and the level accessible to the psychotherapists and psychoanalysts and so on, which is the high level of the personality. Everywhere in between are very important somatic intelligences that we now can target using techniques that are available at different levels of behavioral and cognitive sciences. And actually AI, we're using AI as a translation interface to help us decode the messages and then talk back and forth to these other systems, including the development of some robotic, basically robot scientist platforms that do the experiments according to the various hypotheses formed by the AI, and try to refine these policies so that we can actually make use of all of the capacities that are present in the material so that we don't have to micromanage them. So where modern standard medicine is largely here, these bottom-up kind of techniques focused on the hardware, there's a huge sort of ocean of opportunity opening up in novel applications of top-down control methods. We didn't talk much about this, but basically training cells and tissues to specific outcomes, agential implants like anthrobots in the body that can help heal, and lots of, this is what I mainly talked about, is electroceuticals, which are a subset of morphoseuticals, that is messages that can talk to the material about new shapes and new forms and functions, and all of the details are in these papers. But the bottom line is that I think future medicine is going to look a lot more like a kind of psychiatry than it's going to look like chemistry. The bioelectrical interface is critical to show us how to do that. There are no doubt other interfaces, of course, but the bioelectric one is really powerful, and I think we can hack it for some transformative applications in health. So I'll stop here and just thank the postdocs and the students who did the various work that I showed you today. Lots of collaborators that work with us, and some amazing technical support. All the foundations of different kinds that funded us. 3 disclosures to say here are 3 companies that have spun out of our lab that are funding some of this work. And as always, most of the credit goes to the model systems that actually do all the hard work and teach us about these things. So I will stop here. Thank you very much.